We're going to continue on with antibodies in this lecture. So antibodies are proteins that are made by B lymphocytes. When the B lymphocyte finds its match and gets activated, it divides and forms effector cells called plasma cells. So the plasma cells are the ones that make the antibodies. Another name for antibodies are immunoglobulins, also abbreviated Ig. And these antibodies are very specific to which pathogen they're going to be binding to. The antibodies are going to be binding to the pathogens that are outside the cells in the body fluids. So it could be in the blood, the tissue fluids, or on the mucosal membranes. Antibodies consist of 4 polypeptide chains. There are 2 heavy chains and 2 light chains, and they're joined together by disulfide bonds. Both of the chains contain 2 different regions. There's the constant or C region. That's going to determine the antibody class. So the C region determines the antibody class. So that's whether the antibody is an IgG antibody, IgM, IgA, IgD, or IgE. The variable or V region of the antibody is what binds to the antigen. So the V region binds to the antigen. Here's the antibody structure. You can see that there are 2 heavy chains, and then on the outside we've got 2 light chains. The variable or V regions on the tips of the Y's here, that's what's going to bind to the antigen. And that will vary depending on the antibody because they're all very specific to what they bind to. And then the C region is the constant region, and that determines the antibody class. So for example, all IgG antibodies will have the same C region, whereas IgA antibodies will have a different C region. So a couple of other terms for antibody structure. The stalk of the Y is also called the Fc fragment or crystallizable fragment. And then the arms of the Y are called the antigen binding fragments or Fab fragments. So here's the Fab fragments. So they're on the ends of the Y here. That's what's going to bind to the antigen. And then the crystallizable fragment or Fc fragment, that's going to bind to the white blood cell. So you can see over here we've got a natural killer cell finding a cell that's infected, coated with antibodies, and it will bind to that Fc region of the antibody, and that'll tell the natural killer cell to kill the cell. The Fc component of the antibody can also bind to complement proteins. And remember, that's one of the ways to activate the complement system. There's 5 different classes of antibodies. So star these will definitely be on the exam. So IgG antibodies. They're the most common antibody in the blood. They're the second antibody produced by the plasma cells. IgM are first, but we'll take a look at that later. And the really important thing about the IgG antibodies is that they're able to cross the placenta. So when mom is pregnant, she will pass her IgG antibodies to the baby before the baby is born, and that will protect the baby for the first 2 months of life. The IgM antibodies are big. They are 5 antibodies joined together, so they call that a pentamer. And they're the first antibody that's produced by the plasma cells. The reason they're produced first is because there's lots of binding sites on the tips of those Ys that they can bind to lots of the pathogens. And then once we clean up a lot of the pathogen, we'll switch over with the plasma cells and they'll produce the IgG antibodies, which are smaller and able to get into the nooks and crannies of the body. The IgA antibodies are also called secretory IgA. So these are going to be secreted across the mucosal surfaces. So they're going to be found in the tears, the saliva, breast milk, mucus. They are 2 antibodies stuck together, so they're a dimer. So this is important after the baby is born. When the mom nurses the baby that first milk, the colostrum, is full of IgA antibodies that mom has made, and that will get passed to the baby and will also help protect the baby for the first couple months of life. Here's a look at the IgA antibodies being secreted across the mucous membranes. So you can see that the IgA antibodies, they will be in the mucus. So maybe, for example, this is the GI tract. And the IgA antibodies will bind to the pathogens and prevent them from being able to bind to the receptors that are on our cells and get inside. The next antibody class are IgD antibodies. They're the receptor on the B lymphocytes, so that's what the B lymphocyte uses to find its match. The IgE antibodies are just a single antibody, and they're involved in allergies and also killing parasites. We'll take a look at their use in allergies when we get to chapter 19. And then let's take a look at how the IgE antibodies help killing parasites. So here we've got an intestinal worm. It becomes coated with the IgE antibodies. That's like a big red flag for the eosinophils to come over. And the eosinophils bind to the IgE antibodies, and that stimulates them to release their granule contents, which they contain some nasty chemicals that poke holes in the worm, and then water rushes into the worm, and then the worm explodes and it's dead. And then this is just a nice table that compares the 5 different antibody classes. So you just need to know what we talked about. That'll be on the exam, but some of this other stuff we didn't talk about, so don't worry about it.